Hello. So here we are. We're going to break this quiz 4 down. This is the second part. So we left off with question 16 right here. A restriction enzyme digestion, that means digest means to cut up with restriction enzymes, contains which of the following reagents? Reagents are ingredients. DNA, water, DNTPs, and restriction enzyme. So you know what DNA and water, and you know what restriction enzyme is, but you don't know what DNTPs are. That stands for deoxynucleotide triphosphates. And that's just A's, T's, C's, and G's, adenines, guanines, cytosines, thymines that are free floating. They're inorganic. They're all by themselves. So you don't need those for a restriction digest. What do you need free floating A's, T's, C's, and G's for? That's right. You would need that for polymerase chain reaction. So we don't need that, but we do need the other things. We need water, we need buffer because enzymes require a certain pH and salt concentration. So this is our right answer right here. Booyah. Place the following steps of a general protocol for genetic engineering in the correct chronological order. Well, I'm going to show you guys what that means. So let's say that this is genomic DNA for a mouse. We were going to actually do this experiment in real life, so I'm just going to have to show it to you real quick otherwise. Let's say this is a gene for an enzyme that the mouse makes. And let's say that this gene could be used as a medicine somehow, but we don't want to grow up millions of mice and squeeze the enzyme out of them. We'd rather like squeeze the enzyme out of some bacteria or something. So what we could do is we could get this out of here, and we could do that with a restriction enzyme. We could do it with PCR. We'd probably do it with PCR, to tell you the truth, and we'd probably do it with reverse transcriptase PCR. But let's not talk about that. Let's just talk about getting this out. So what you need to do is cut that out. And we would probably do it with PCR, although you could do it with a restriction enzyme also. So once you had that gene for that enzyme of interest, what you need to do is you need to get it into a vector. And a vector means a little vehicle that we can use to get a gene from one place to another. And we use plasmids as vectors in our class. You could also use viruses as a vector, but we're going to use this plasmid. So what we would do is we would choose, just like one of those other questions, we would choose a restriction enzyme that makes a cut in this multiple cloning site. And we would use the same restriction enzyme to cut here and to cut here. And what that would do is it would make it so that once our vector was open, it would make it so that this end of this gene hybridized with this end of that. And then we get it into our plasmid. Once we have it into the plasmid, then we can put the plasmid into bacteria. And we'd need a selectable marker and all that. And then we could make 1 billion copies of this bacteria or this yeast or whatever this slave organism is that we're going to use to make a whole bunch of that for us. So what would happen is this bacteria would go and it would read that copy of that gene in order to make the enzyme that we want. And let's say that's the enzyme that we want. Once we have 1 billion of these cells making 1 billion of these enzymes, we can murder all of the cells and we can use chromatography to separate that enzyme from all the rest of the cell guts. So that's how we can make medicines, biopharmaceuticals, through gene cloning, or taking a gene out of one organism and putting it into another. So this cell right here, this cell right here is called a recombinant cell because it has mouse DNA in it. So this cell is recombinant. We made a recombinant cell line. Okay, so anywho, Place the following in order. First, we're going to need to isolate the gene of interest. Next, we're going to need to get that gene of interest, paste it or clone it into a vector, into a plasmid. Next, we're going to need to get that plasmid into host cells. So that's going to be a transformation that we're going to do that with. Transform host cells with the vector. Next, we're going to grow those recombinant cells. And then we're going to murder them, and we're going to use chromatography to purify that product of that gene. Boom, like that. Okay. When performing a transformation, ice-cold calcium chloride makes the cells chemically competent. What that means is that it makes little holes inside of the cell membrane, cell wall, through which the DNA can enter. So they make it more permeable to DNA. Which of the following techniques should be used to amplify, that means make lots of copies of, specific DNA sequences? Surely that would be PCR. But let's go over these because these might be in future questions. PAGE is polyacrylamide gel electrophoresis. This is the same gel electrophoresis we do normally, except it's with proteins instead of with DNA. This separates proteins according to size. A Bradford assay, we did that a whole bunch in applied chemistry with the milk, you might remember, and we were able to use a Bradford assay to quantify or measure how much protein there is in a solution. PCR is polymerase chain reaction. ELISA is the antibody test that's used to test for a certain protein, if a certain protein's there or not. So anyway, PCR for this one. Place the following steps in order of a PCR cycle. So first, you need to use heat to denature the double-stranded DNA into single-stranded DNA. Then the primers are going to hybridize. That's going to happen at a lower temperature. And then we're going to bring it up to the temperature of that Yellowstone hot tub in order to use DNA polymerase to extend the primers and make copies of the DNA. Booyah. Okay, match the type of chromatography to the property it uses to separate proteins. You guys don't know anything about all these different types of chromatography. Really, you don't think you do, you do. But it doesn't matter. We're talking about test-taking skills right here. We're not talking about a knowledge test. We're talking about a skills test. So anyway, chromatography means separating things out according to chemical or physical properties of the things, taking a mixture of a whole bunch of stuff and getting one thing that you want, all purified all by itself. So gel filtration / size exclusion chromatography. Well, look at this. Size, booyah. We don't need to see any more. Ion exchange chromatography. You know, ions are charged, so we look for something with ions or charge. Boom, charge, boom. Next, affinity chromatography. Affinity means attraction to. And you guys did this with the histidine tags. You remember that? We purified one of those proteins with chromatography using histidine, nickel beads and histidine tags. So anyway, that relied on the presence of a unique functional group, like that polyhistidine tag functional group. And then hydrophobic interaction chromatography. This has to do with whether things like oily things or watery things better. And that separates proteins based on how much they like water or their solubility in water. Booyah. Okay, question 22. Many biopharmaceuticals must go through a purification process to separate them from all the other junk that was in the cells. Which of the following is commonly used to separate proteins from a bunch of cell junk? Well, that would be Chromatography. Yeah? Chromatography. That's what it does. Okay, a technician is asked to verify, that's to see if it's there, the presence of a protein of interest after a production run. So he does SDS page. So he runs a protein gel and then a band appears, and the band is where it's supposed to be. But how can he be sure that it's the right protein? Well, he could do ELISA to see if it's the right protein, because he could use an antibody that only sticks to that protein. And when ELISA is done on a gel, ELISA is called a Western blot. I know, I'm so sorry, but it just is. It's called a Western blot when ELISA is done on a gel instead of in a test tube. Yeah? Will you take my word for it? Okay. Diagnostic immunoassays, so they're talking about ELISA again, for infectious diseases such as strep throat or COVID-19 are commonly used by physicians to quickly diagnose patients. These immunoassays utilize the reaction of antibodies to which of the following from a patient's sample? That would be antigens. So, like the spike protein in the case of ELISA, in the case of ELISA testing for coronavirus. Please refer back to that video on testing of coronavirus. You're going to want to watch that again at this point. Place the general steps of generating a monoclonal antibody in order. Well, to tell you the truth, I'm going to have you guys watch this video right here. I'm going to link it to explain monoclonal antibodies. I usually think that I can explain things better than other people, but I think that this dude explained monoclonal antibodies way better than I could in 4.5 minutes. So watch this video for this question, but here's the answer. First, you're going to want to inject an animal with the antigen that you want the antibodies to be against. So that's what you do first. And this question's kind of weird. We're going to repeat. We're going to inject that sucker again. So wait a couple weeks, do it together. It's like a booster shot. You ever had a booster shot for immunization? That's what it's all about, booster shot. After that, they don't do this to you, but they're going to do this to the mouse or to the rabbit. They're going to collect some blood serum. They don't have to kill the whole thing. They just bleed out a little bit of it. Collect some blood serum. After that, they're going to isolate B cells or beta cells. That's a kind of white blood cell that makes antibodies. They're going to isolate those cells from the blood. After that, they're going to fuse those beta cells with tumor cells, because B cells don't grow very well, but tumor cells do. So if you create what's called a hybridoma, which is sort of like Frankenstein between a white blood cell and a tumor cell, then you have a white blood cell that makes antibodies and it divides like crazy. And then in the Petri dish, those cells are dividing like crazy, and you can isolate cells that are making the correct antibody and then just grow a bazillion of those. And then you can use chromatography in order to separate those antibodies from all that other cell junk. And then you can use it as a drug or a diagnostic tool. Booyah. That's what's up. Okay. Oh, yeah. By the way, yeah, that's hard. But remember, you're not here to get all these questions right. You're here to pass this test, because the way this test works is you either pass it or you don't. There's no A, B, C, D, F. There's pass or don't pass. And you want to pass it. You don't have to get them all right. You want to get lots of them right, though. The difference between a direct ELISA and an indirect ELISA is that the conjugated enzyme to be detected is linked to a primary antibody in a blank ELISA, whereas the detection enzyme is linked to a secondary antibody in a blank ELISA. You don't have to know crap about antibodies or ELISA. Just read between the lines. They're talking about direct and primary. What's primary mean? First. And they're talking about indirect and secondary. And what's secondary mean? Second. So surely the primary antibody must be associated with direct ELISA, and the secondary antibody must be associated with indirect ELISA. Test taking skills. Better act, somebody. Both antigens and antibodies can adhere, that means stick, to the wells of ELISA plates. That's the little test tubes I talked about in that video on how COVID-19 ELISA testing is done, which you should watch again, through non-specific binding to plastic. So it's saying, can random proteins stick to the plastic? Yes, they can. Which type of ELISA is the most appropriate to determine the concentration of a specific antigen in a sample? Here's the key word, concentration. That means how much. So they're not talking about is it there or not. They're talking about how much is there. Qualitative means is it there or not. Quantitative means how much is there. So if we're talking about trying to measure concentration, we're trying to do a quantitative test. Again, that's a test taking skill. You got to know what those words mean, though. Quantitative, quantity, how much. Question 29, sandwich ELISAs. Check it out. Here's how a sandwich ELISA works. You take an antibody that, remember, these plastic wells, antibodies or proteins, it's just going to stick to the plastic. Then you attach your antigen, and your antigen's only going to stick if that antibody's there, yeah? Then you add another antibody that is also designed to stick to that antigen, and it makes a sandwich. See that? It's antibodies for bread and an antigen for the peanut butter and jelly. Then this second primary antibody, primary because it sticks to antigen, it doesn't stick to an antibody. An antibody that sticks to an antigen is a primary antibody. An antibody that sticks to another antibody is a secondary antibody. Anyway, the second primary antibody gives off some light so you can see that it happened or not. Yeah? Yeah. Okay, sandwich ELISA. So an antigen must be recognized by both a primary and secondary. No. Antigen must be recognized by two primary antibodies to give a positive result. Yes, that's right. Which of the following is typically measured using a spectrophotometer set at 600 nanometers? 600 nanometers is the wavelength of light that bacterial cells party at. So this would be used to measure the concentration of bacterial cells in a liquid culture. So not on a plate, in suspension. That means in suspension, like suspended in liquid. So the concentration of of bacteria. If it was DNA, we'd have to use 260 nanometers. 600 nanometers for bacteria, 260 nanometers for DNA, 280 nanometers for proteins. That's what wavelength those different things party at. Question 31. Preventative maintenance and calibration on laboratory equipment is scheduled according to which of the following? When the manufacturer tells you to calibrate that pipette, do it. Yes. Based on how often the equipment is used. You're using it all day, every day, you better do it more often than if you're using that sucker 5 times a year. Based on the company's standard operating procedures for preventive maintenance and calibration. Yeah, you do it when your boss tells you to. So all the above, booyah! How is this number correctly written in scientific notation? Well, it's way smaller than 1, so it's going to be to the negative power. And there's 1, 2, 3, 4, 5 orders of magnitude displaced to get the decimal point to right here. So that would be 5.2 times 10 to the negative 5. Booyah. What is this number rounded to 3 significant digits? Leading zeros are not considered significant. This one's significant, this one's significant, this one's significant. 3 significant digits would be 0.986 because this 5 would round to a 6 based on the 7 right here. So 0.986. Booyah. Submit the quiz. How do you think we did? I hope we didn't get any wrong. Oh. Did we get them all right? Did we get them all right? Yes, 38.5 out of 38.5. We got them all right. We're going to win. We're going to be winners. I don't know why. Because we're bosses.